Hello everyone, I'm Anuj. Thanks for being here. I'm filling in for my advisor, Jean-Pierre Leburton, who's currently out of town, unfortunately. So before I begin, I just want to say that I'm battling a little bit of a cold, so if you can't understand what I'm saying, please interrupt me, and I'll try to be more clear. So to start, I want to just remind you of this current state of semiconductor technology. We are currently able to produce multi-layers with individual layer thicknesses below 1 nanometer. So what does this mean? Well, if we're trying to sequence a DNA molecule, that's getting a very thin layer feature size is very important, as the nucleotide spacing is on this order. So here is a multi-layer silicon device with, multi-layer structure with a 0.7 nanometer oxide layer. So just to show the actual scale of what we can feasibly fabricate. In addition to that, we are able to create nanopores in silicon on the order of a few nanometers with e-beam lithography. So as the diameter of a DNA molecule is just that size, this is perfect for these kinds of applications. So a little background on DNA sequencing history. So almost a decade ago, people were trying to pass a DNA molecule through a nanopore and actually pass an electron current through each nucleotide. So the idea was is that the current would be a specific function of which nucleotide they were going through. And so if we analyze the current, we can hopefully sequence the DNA that way. But unfortunately, this turned out to be a little bit difficult, so other methods were sought out. So one of the more popular methods, which is still actually being used today, is the Ionic Blockade current method. So what happens is we have a solid state nanopore, just a nanopore in a solid material, and we actually pass a stream of ions through that nanopore. So if we actually pass a DNA strand through the nanopore as the ions are flowing through it, we should see a decrease in the ionic current. And that is actually precisely what we see. So every one of these little dips is an event where the DNA is actually passing through the nanopore. So I'll speak more on this in a bit. Now, the thing is with these methods, the nanopore membrane is a passive membrane, meaning that it only serves to create the hole which the ions go through and passively, passively interacts with the DNA molecule. So our idea is maybe we can make a multi-layer device that can actually manipulate the electrostatics and the behavior and motion of the DNA molecule within the nanopore. So a lot of efforts have been done here and with other groups. So a little bit more on the Ionic Blockade method. So essentially what happens is when the DNA is through the nanopore, the ionic current through it is decreased a bit. So depending on the actual orientation of the DNA molecule within the nanopore, we see a specific signature in the blockade current. So a larger current that occurs for a longer time stretch is a signature of a unfolded DNA strand, whereas a folded DNA strand should block more of the nanopore and hence have a smaller current. So with this method, we see an inverse relationship with the nanopore diameter or a inverse quadratic dependence depending on the actual thickness of the membrane. But the point is that this is indeed due to the current blockade. As we increase the nanopore size, we should have a lower ionic resistance. So here's an example now of a device which is not a passive device, an active device. This is actually an older schematic, but it serves this purpose well. So it's a multi-layer device. It's coated with a metallic film. And by applying different voltages to these layers, hopefully we'll be able to tailor the electrostatic profile in the nanopore to manipulate the DNA. So one example of a multi-layer device is a simple PN junction. So a PN junction is just is two layers of silicon, one which is doped with extra negative charges and another one which has extra positive charges. So if we hook up each layer to a voltage source, and if we apply different voltages to each layer, we can actually change the charges that are present within each layer. As a result, we should change the electrostatic profile within the nanopore itself. So one main result is if the nanopore is in an electrolytic solution, we should have an accumulation of different charge species around each layer, depending on the actual voltage of the layer that we're talking about. So these plots show 4 different bias voltages on these 2 layers, and we can actually see that the ionic current, or excuse me, the ionic concentration within the nanopore is heavily influenced by the voltages which we apply to these layers. So in one extreme case, we have a sort of ionic monopole, if you will. It's a symmetric charge distribution of the ionic species. But if we actually change the voltage profile, we can get a sort of ionic dipole. So this just shows that we can actually manipulate the electrostatics within the nanopore by using this formalism. And I should mention that this is very similar to how biological nanopores actually function. So we're getting a little bit closer. So one application of this type of electrostatic manipulation is a diode-like current filter that just based on the voltages which we apply to each layer. So this blue curve is an ohmic current curve. So no matter what the sign of the bias is, we have a linear relationship with the bias. However, for specific voltages, we can actually make a diode, meaning that one bias sign is preferred over the other. So what this means is actually we can control the direction of the current just based on applying a voltage to the nanopore. In addition to that, we can actually filter out one ion species versus another just based on this electrostatic manipulation. So depending on the actual electrostatic profile, we can choose chlorine ions only going through the nanopore or potassium ions only going through. So what you should take away from this is that we can actually tailor the electrostatic profile to a strong degree just by changing the voltages of each layer. Now, this can actually, we translate this to manipulating the DNA as follows. So depending on the actual voltage profile within the nanopore, as the DNA molecule is being translocated through it, it can either be translocated as a crumpled ball or it can be translocated as a long stretched strand. So this red curve essentially measures how much stretching there, these red and blue curves measure how much stretching there is in the DNA. So this red curve corresponds to a DNA molecule that is elongated being passed through, and this blue one corresponds to a crumpled up ball. So just by manipulating the voltages on each of these layers, we can actually control the shape and orientation of the DNA molecule as it passes through. This is a plot of the translocation time as a function, the translocation probability as a function of time. So this blue curve is the crumpled form of the DNA passing through quickly in one shot, and these red events are the elongated DNA taking a longer time to pass through. Okay, so we've just shown that we can indeed change the electrostatic profile within the nanopore just by applying voltages to each layer. So now we want to see, is there a different way to actually sense the DNA being passed through? And graphene turns out to be the perfect material for this application. So the reasons are primarily the thickness of graphene. So it is only 1 atom thick. So that means that we can actually sense each nucleotide at a very high resolution. Normal solid-state nanopores are at least a few nanometers wide. So the resolution is limited to a few nanopores, or a few nucleotides. But with graphene, we can actually sample on the individual nucleotide basis. In addition to that, graphene is electrically active, meaning that we can pass a current through it. We can control the carrier concentration within graphene. So hopefully by using graphene as the nanopore membrane, we can do some additional tricks to try to sense this DNA. So one way we can attempt to manipulate or influence the DNA is by actually placing charges on the mouth of the nanopore. So the DNA molecule is composed, the backbone of the DNA molecule has a bunch of negative charges. So presumably, if we put negative charges along the nanopore, this should hinder the flow of the DNA. So here's a little movie showing this simulation. So here we have a negatively charged hole, which is seemingly slowing down the flow of the DNA through the nanopore versus the positively charged hole. So the actual mechanism for this is twofold. First is the actual electrostatic repulsion or attraction between the charges. But even in addition to that, if we have a negatively charged pore, we actually see a higher current of potassium ions flowing vertically upwards through the pore. So this actually creates a drag force, if you will, on the DNA molecule, which slows it down even further. So this is one way to actually manipulate. This is another way besides the electrostatic manipulation of the nanopore to control the DNA. So hopefully we can combine all of these technologies into a multi-layer device to simultaneously control and sense the DNA molecule as it's being passed through the nanopore. So here's a very simple schematic of a graphene layer, it's kind of hard to see, which is sandwiched between two oxides to prevent interaction with the solution. And down here we have a back gate, which we will apply a voltage to manipulate the charge profile within the graphene sheet. So before I continue, I also want to mention the unique electrical properties of graphene. So as some of you probably know, the graphene nanoribbon is just a slice of graphene. It's a pseudo 1D strip of graphene. And so depending on how you actually cut the strip, we can have either armchair edges or zigzag edges. And the peculiar thing about graphene is that the actual band structure, or the electronic structure inside the molecule, is strongly dependent on the edge shape. So here's two diagrams of the band structure of a graphene nanoribbon with a zigzag edge right here and with an armchair edge right here. So as you can see, the electronic profile is wildly different depending on the edge. So maybe, actually, if we take a graphene nanoribbon with a complex edge, perhaps we can push the electronics to a degree which will create a lot of sensitivity and interesting effects as the DNA is passed through. I also should mention that in this model, what we try to do is we pass a current from this source to this drain through the graphene sheet. And as the DNA molecule passes through the graphene nanopore, hopefully there will be some interaction, some electrostatic interaction between the molecules of the DNA and the graphene mouth, such that the current is manipulated as the DNA is being passed through. So hopefully we can extract some information from this and thereby get a sense of what is going on with the DNA. So this leads us to the idea that if we perhaps make a quantum point contact, which is essentially a small constriction within a longer device, then maybe the interactions of this edge and the electrons will create some sort of very complex, very nonlinear current curve as the DNA is being passed through, such that we can actually extract some information. So I'll go a little bit more into that. So, like I said, the edge of the graphene nanoribbon is strongly connected to the electronics. And the reason is because the current is dependent on this quantity called the transmission coefficient. So this is essentially the probability that an electron will hop from one end of the nanoribbon to the other. So as we change the edge of the graphene nanoribbon, we strongly change this transmission coefficient and hence the current. So here's an example of this in action. So we have 4 different types of graphene nanoribbons. This first plot is of a 5 nanometer wide, pure armchair edge graphene nanoribbon. So there's no shaping done to it. This one over here is a 15 nanometer pure graphene nanoribbon. And then these two are an 8 nanometer and a 23 nanometer QPC edged nanoribbon. Now, within each specific geometry, we actually have 4 pore configurations. So the black curve corresponds to no pore present. The blue curve is a 2 nanometer pore present at the point P. The red curve is a 2 nanometer pore present at point Q, and the green curve is a larger 4 nanometer pore at point P. So let's focus on this plot right here. So the black curve is a standard staircase transmission coefficient from an armchair nanoribbon. Nothing too crazy happening here. But as soon as we add a nanopore in the center, we actually see a very strong change in the transmission coefficient. So just the removal of a few atoms in the center of the nanoribbon can wildly affect the electronics of the nanoribbon. In addition to that, actually changing the position of the nanopore introduces large changes as well. So it's clear that introducing these sort of lattice deformations to the graphene nanoribbon can cause very, very complex interactions with the electronics. Now, why is this important? Well, like I said before, the current is dependent on the transmission. So that means that, well, this is the conductance actually, which is related to the current. But that means that depending on the actual geometry of the graphene nanopore, we can get very interesting behaviors of the conductance. So here we see the conductance as a function of the Fermi energy. The Fermi energy is essentially the energy, the base energy of all the electrons in the graphene nanoribbon. And this energy can be thought of as the voltage that we apply to this backgate. So as we change this voltage, we change the Fermi energy of the electrons in the graphene sheet. So let's take a look at this plot right here. So we see that at a very low gate voltage, or Fermi energy, nothing interesting really happens. As we increase the gate voltage, the current increases, but in a strongly nonlinear fashion. So let's focus on this range right here. So if the Fermi energy is in between these values, we actually see a very strong increase in the current over a small Fermi energy range, as opposed to, say, this region, where the current does not change very much as the Fermi energy is changed. So why is this important? Well, the Fermi energy, like I said, is a function of the voltage of the backgate. But in addition to that, it's actually a function of the electrostatic potential of the entire environment. So if a DNA molecule is being passed through this nanoribbon, perhaps the molecules of the DNA can change the Fermi energy locally around the nanopore and actually manipulate the current within the nanopore. So the idea is, as the DNA is being pulled through, the conductance or current will actually be changed. Now this is important because if we can actually tune the graphene layer to be in a regime where it is highly sensitive to changes in the Fermi level, perhaps then we can see large changes in conductance as the DNA molecule is being translocated. So in this scheme, to test this idea, we actually place a single charge within the nanopore and see how the current is conductance, is changed. So in this case, we actually show that we see fluctuations of the conductance on the order of a fraction of a microsiemens, which is a very detectable amount with today's technology. So depending on the actual Fermi level within the graphene sheet, the change in conductance can be quite large, especially for the wider graphene QPC. So let's say now we have a specific device and we don't see something as the DNA is being passed through. Well, we could create a new device that is more sensitive, or we could actually change the Fermi level such that the device does become more sensitive to electrostatic changes. And this is important because the Fermi level can be changed in the lab just by tuning a knob. We don't have to create a new device. We don't have to do anything like that. So we can actually tune the sensitivity of our device just by changing one parameter. So now here's the meat of this talk. So let's take a DNA molecule and pass it through this nanopore. As a function of time, the electrostatic profile of the charges on the nanopore will change over time. So the reason why we see this rotating is because the DNA is a helix, and every time and this is the actual electrostatics within the plane of the nanoribbon. So at every snapshot, we see the DNA sort of rotating in place. So these are the actual same 4 geometries that we talked about earlier. And this is the conductance in the graphene sheet as a function of time as the DNA is being translocated. So each of these curves within each box, it corresponds to a different Fermi energy, just to show how the actual variation in the fluctuations as the Fermi energy is changed. So let's take this is the 15 nanometer QPC, 8 nanometer QPC as an example. So if the Fermi energy is very low, this is about 0.04 electron volts, the actual fluctuations in conductance as the DNA molecule is passed through is very small. It's only about 0.6 microsiemens. But if we actually change the Fermi level to 0.16 eV, we actually see the conductance increase to almost 3 the conductance fluctuations increase to almost 3 microsiemens. The effect is even more enhanced over here. For a very low Fermi energy, we see almost 10 microsiemen fluctuations. And for the highest Fermi energy, we see a smaller 3 microsiemen fluctuation. Now, this does seem a little confusing at first. You may ask, why does increasing the Fermi energy decrease the fluctuations? Well, if we look at our conductance plots, it actually is not a linear relationship between Fermi energy and conductance. For example, take this case down here. We can actually increase the Fermi energy and decrease the conductance value. So there's not a strictly linear relationship between the two. So the point is that we can actually extract some very interesting behavior and enhance the interactions between the DNA and the conductance just by tuning the Fermi energy, in addition to tuning the geometry. So we actually have multiple control knobs to the sensitivity of our conductance to the DNA translocation. Now, putting all this together, hopefully we can create a multi-layer device that has a sensing layer where we can actually sense the current on the DNA. Perhaps another layer which can act as an additional gate or something that can manipulate the electrostatics within the nanopore to change the length of the DNA, the conformation of the DNA, how fast it's going through, where in the pore it's being positioned. In addition to changing the actual electronic profile within the sensing layer via a backgate. And the possibilities are endless. The only thing now we have to do is try them out and see what kind of results give us what we want. So these devices are actually being created here at Illinois by Rashid Bashir. He makes multi-layer devices with gates. And we are doing an ongoing collaboration with him to see what we can get out of this. So in conclusion, semiconductor membranes are extremely flexible. We can manipulate them just by changing the voltage on each layer. So we can actually tailor the electronic profile within the nanopore. Graphene is extremely thin, so we can access the DNA on a single nucleotide resolution. It's electrically active, so we can manipulate how the current interacts with the DNA. And it's very patternable, so we can get very exotic edge geometries to create, hopefully, a very sensitive device. So putting all this together, we can get a QPC transistor, which the carrier concentration can be controlled by a backgate, and hence the sensitivity of the device to external charges. And so finally, hopefully this will all come together into one very nice multi-layer QPC transistor, with many tunable knobs to increase the sensitivity of our device. So.
Thank you.